So I wanted to structure it in two little parts. First one is a biological teaser, and I know that this is an Institute of Applied Mathematics, but I wanted to really show how we can get the fact that we can get to high resolution for a real life protein. And then in the second part of my talk, which would be larger, and obviously we can maybe have more discussion on that, I would show what exactly we do and how we do this and how we think about subtomogram averaging and tomography. So we're interested in membrane proteins. Membrane proteins are interesting because they're major drug targets, and almost over half of drug targets in the modern market are targeting membrane proteins. But it's very hard to get the structures out of them, mostly because historically they've been more difficult to purify. And structures of membrane proteins are generally hard to produce because we first need to extract them from the membrane. And then, those structures can be used for drug design and to understand how these proteins function, which is also important. But what my lab, the biological focus of my lab is how lipids regulate structures of membrane proteins and also the other factors such as gradients across the membrane. And for this, we generally use three methods. One is we do biochemistry, we purify membrane proteins and then do single particle cryo-EM, 2D imaging, and then do 3D reconstructions. This does give us very high resolution structures, but essentially this has limitations that you need to purify your protein first. And then tomography of native membranes, and we do that in order to find our protein of interest. We focus on ion channels, and then if we can find our proteins of interest, we can do subtomogram averaging. So we had several talks on subtomogram averaging today, but I will not go into too much details. But yeah, essentially our group developed methods. We mostly do them for ourselves, but then whatever we find useful, we also release for the community. And then some of the people actually really find it super exciting to release methods. And so we have a GitHub where we release something that is ready to be used by someone else. Okay, so I want to give you one example of what we do. It's a protein that is a part of the excitation-contraction coupling in skeletal muscle. Don't be afraid of this. It's actually very simple. So a motor neuron sends a signal to a skeletal cell, this generates an action potential that goes. It's an electrical excitation that goes on the surface of a cell, and then at some point it gets into an invagination in the skeletal cell that's called T-tubule. So electrical signal activates voltage-gated calcium channels activated by voltage, and calcium can penetrate through. And so there's usually 4 voltage-gated calcium channels, and 4 of them are attached to one even larger calcium channel that's called the ryanodine receptor. So when calcium goes inside the cell, and the physical interaction between the voltage-gated calcium channel that's called DHP receptor and the ryanodine receptor leads to opening of the pore of the ryanodine receptor, RYR1, in skeletal muscle, and then even more calcium can be released from the sarcoplasmic reticulum, which then leads to the contraction of the actin myosin fiber. It's a very exciting system, and the structures of the purified components have been reported. So obviously DHP receptor structure has been reported. There are several structures of ryanodine receptor that have been purified and then either solved in presence of detergents around the transmembrane domain or lipid mimetics were used. But so one way or another, these are high resolution structures, and they have been reported roughly 5 years ago. Interesting thing about ryanodine receptors, they're a little bit different in the heart and skeletal muscle, is that they form clusters, and so every second ryanodine receptor has DHP receptors attached, every second one doesn't, and so they make a little checkboard. And then when they are in native membrane, they obviously also regulate each other's function. And then once you purify all those, you will lose a lot of those interactions, and also you lose the native lipid composition that is the sarcoplasmic reticulum membrane. So what we wanted to do from a biological standpoint is to try to understand the structure of the supercomplex And then once we can do structures, we can then also induce different functional states and understand the activation mechanisms of the supercomplex. And the conformational landscapes, which we hypothesize, would be slightly different from those that are in the purified receptors. And then also regulation in the clusters is also very interesting. So for this, Wenbo Chen, who was my student in Frankfurt at the Max Planck for Biophysics, optimized the existing protocol, which have been used previously by several groups. It's a freshly sacrificed rabbit. We get the muscle, meat from it, and then there is a few steps of homogenizing centrifugation, sucrose gradients, which essentially allows us to get fractions of the rabbit muscle, and then one of them, this fraction over there, and the last sucrose gradient contains both ranitidine receptor and the DHP receptor. So we checked it with biochemistry, but essentially we don't purify the protein here. We purify membrane fractions that are that contain those proteins, and then we put them on the cryo-EM grids and image it. So this is how it looks like. So this is a tilt series, right? I mean, you've seen a lot of tomograms of tilt series. We use the gold beads to align them, and then these are little vesicles. You don't see anything in the tilt series themselves, but once you perform a 3D reconstruction, so this is a slice through a 3D reconstruction, you see large membrane proteins over here, which are ranitidine receptor. They are roughly 40 nanometers in the widest dimension. And you can see them by eye. It's very, very, very easy to identify the tetramers as they should be from the different direction if you look from them from the top. And then in some cases, we could obviously see that there is an extra membrane, which would represent the T-tubule. But actually, to be honest, most of the receptors were freestanding and only 10% were attached to something that looked like another membrane. So we couldn't do a very high resolution structure of the receptor with a T-tubule yet, but I hope that we can do it in the future. But we, in our first analysis, we could do a structure at roughly 13 angstrom. 13 angstrom is not is high enough resolution to actually be able to fit the rigid body, to fit the available atomic model using the domain structure, and the fit actually is very good. Over here, we can see that the structure solved by the lab of Joachim Frank fits rather well. Usually they draw the membrane horizontally, but here the membrane was curved in a non-trivial way. So the curvature of the membrane was actually higher than of the vesicle in which the protein was present. And we found, you can also see this in a tomogram, for example. There is a membrane, and then next to the receptor, it has a little invagination. So it's very easy to replace the buffer. So we added a ligand that's supposed to lock the receptor in the constantly open state. It's called ryanodine, after which the receptor is also named. And so this is a toggle between the structures is of lower resolution. So this is a toggle between the structure in the presumably closed state without any ligands and the open state. And we see that while the outer domains move similarly to the structures when they're purified, the membrane also is deformed. This is interesting and non-trivial, and this means that once the receptor is open, there is some amount of elastic energy stored in the membrane, and then when the receptor is open, the membrane would want to obviously bend back, and it would probably favor closing of the receptor a little bit faster than if there would be no membrane. So 13 angstrom is a high enough resolution, but it's in a way the worst resolution that you can get, because you spend a lot of effort getting it, but you don't see a lot of it. And so we spend a lot of thinking and effort into ways to improve this resolution. Our first idea was to do this, what we call hybrid subtomogram average and single particle cryo-EM. So what we do, we record a high dose image, something like 20 electrons per square angstrom when the image is in an untilted stage. So Nico Grigoriev yesterday showed us how from the untilted images it's possible to locate objects, but it has certain limitations like a very large size and limited flexibility. So we tried to pick random receptors from those images, but we didn't get it to work yet. So we do need tomograms to be able to identify the particles. And so after recording a so-called high-dose image, we then record a tomogram with the same optical settings. So, and then we incorporate the high-dose image into the tomogram, and then perform subtomogram averaging as we do usually. So here you can see the structure in B, it's also 13 angstrom. But then what we do, we perform the final reconstruction only from the high-dose untilted image. And when we do that, we're able to refine the positions of the particles in the tilt series against the final structure and get slightly improved resolution. Here we got 9 angstrom, and we can see a secondary structure. So all those helices that you can, all those little sausages that you see are alpha helices, and the protein is mostly made of alpha helices. And in a nice 9 angstrom map, you can actually see them well resolved. And we can also position the high resolution atomic model. And then we can see that there is a little difference already that we can see between the structure of the purified receptor and the structure that is in the native membrane. We were very happy about the structure. And so what essentially we do at this point is that we perform a 3D reconstruction from a limited number, roughly 2,500 particles with C4 symmetry, but of limited dose and of rather thick ice. However, because we could minimize the problem of subtomogram averaging to a single particle structure problem, we were actually convinced that if we get unlimited amount of data, then we will be able to improve the resolution even further. So we first were hoping to get something like 6 to 7 angstrom. And so we kept pushing it, and then the new structure that we actually got in the beginning of this year came already from my new lab, which Vasily only started in October last year. The perk here is that much more data, roughly 4 times more, but we did have an improved hardware setup. We collaborated with Abi from Thermo Fisher Scientific. He runs an application team there, and he was enthusiastic enough to record some tomograms from the grids that we sent him using the new Cryos, a new Falcon, and a new energy filter that Thermo Fisher has. And then we used the methods about which I'll tell you a little bit later in my talk to process the data in a streamlined fashion and perform classification. And Vasily used Reliant. Right now he uses Reliant 4. It does get very pretty reconstructions. And he got the global average of 4.6 angstrom, and he could see very, very nice density for all the helices, but for this one, the long helix in the transmembrane element, looks particularly beautiful. And then he can go for a local refinement that went to 4.0 angstrom, and at that resolution, you can really, really see side chains. That's a very good map. And obviously, we have the atomic model that we can refine into this density map using the sidechain information. So essentially, we can build an atomic map of the ion channel in native membrane. It's not the native tissue, but it's a native membrane. So the composition of lipids, for example, is the same. Neighbors are also the same, and we also see that there are interactions between neighboring receptors in the membrane. Now, one thing that I'm really interested in is was this whole solving the structure worth it? So we performed analysis of the conformations. I'll go into this briefly. So the way that the receptor works is they're in apostate. So apostate means that there are no ligands bound, and the pore is in the closed state. One of the physiological ligands is calcium. It comes in and binds to the calcium binding domain, and it primes the receptor to go into the so-called prime state. It's not yet open, but it's ready to open when another ligand, the physiological ligand is ATP, binds, and then basically the pore gets open, the calcium can go through the pore. And then at some point, the receptor gets deactivated, and the whole circle continues. And then obviously ryanodine locks the receptor in the open state. So what we've done, we took all the available atomic models from high-resolution cryo-EM maps, and we calculated distance to each other. We aligned them and calculated similarity between them. And then we projected this multidimensional space on a 2D coordinate plane. So essentially, most of the covariance was in the first two principal components. So those structures that are similar, close to each other, are similar. Those that are far away are less similar. Blue ones are structures in the apostate, so without any ligands. Cyan ones are primed, and green ones are open state, right? So, and the yellow ones are inactivated. So, yeah, we can actually, in this diagram, we can actually see, a little bit of a conformational transition between priming, then activation, and then activation. So we also put our atomic model here, and then it's in red in situ, with in situ apostate. So we only have one structure at the moment, but what we can see from it is that it's something between primed apo and the open state, which suggests, I can maybe speculate on that the structure of the apostate, at least, is different in native membranes compared to when the protein is purified, and is different enough that you can actually that you consider this close to the open state than to the non-open states on purified receptors. So what we're trying to do now is okay. So the little conclusion here is that there are membrane proteins in native membranes, and they're different to the structure in the native. So basically, structure in native membrane is different to the structure of purified receptors for at least some proteins, and obviously I cannot say about all of them because we didn't look at all of them. And also that it's possible to obtain high resolution structure by cryo-Electron tomography and subtomogram averaging for a membrane protein, which is not a trivial conclusion. So what we're trying to do now from the biological standpoint is to look at the classification of the random receptor, and the subtomogram averaging is a little bit more difficult to classify than single particle cryo-EM data. We heard about this a little bit, and then at 4 angstrom structure, there is a potential that we can actually see small molecules bound to the, for example, transmembrane domain of the receptor, and we explore if we can look into that. So now it took us several years and a lot of thinking to get from 9 angstrom to 4, because while the number difference is not too much, it took us quite some tweaking, and also the entire community also obviously advanced. So when we looked at the structures obtained by subtomogram averaging, and I hope my text is not too small, most of those beautiful structures come from particular classes of proteins, and those are other lattices, right? So they would either form some kind of higher order structure, which is relatively well ordered, for example, COP2 codes or structural proteins of the viruses. They could be in a filament, for example, like in the beautiful papers from Stefan Raus's group, several outstanding papers from Yulia Mohammed and Dmitry Tegunov also have ribosome in it. So what they all have in common is that they all are large or form a lattice, which helps to align the particles. And then Chris Russo went into details about, the potential lower limit about for in situ structural determination. And then Nico also talked about this yesterday. They're all rather abundant. So, like, for example, if you have a lattice in a tomogram, then you pick a lot of particles from one tomogram, and then usually they're well ordered. So our random receptor is only large from all those characteristics. It's not very abundant. We have roughly 40 per tomogram, and then if we want to go for a larger magnification with smaller pixel size, we would have even less of them. It's not very well ordered, and it doesn't go to very thin ice because we look at the vesicles, and they do give us significantly thick ice. And then I also want to say that most of the proteins in human cells are neither large, neither too abundant, or likely not very well ordered. So in order to kind of make subtomogram averaging a little bit more applicable to what we do, but also at the same time more applicable to all the other applications, I think we need to really go into the direction of trying to do structures of other proteins, basically more regular everyday proteins. And for that, one thing that's important is throughput. Tomographic data collection was always very slow, and to record one tomogram, it would take roughly 20 minutes, maybe a little bit longer. And then we know that we have 40 particles in a tomogram. So this means that you can put $1 to every particle that you record. Luckily, Bartesaghi and Danev labs recently, I think, revolutionized data collection field because we can now record multiple tomograms at the same time, and it increases the throughput maybe by an order of magnitude, and then now you can collect roughly several hundreds of tomograms per day, a little bit dependent on whether you can image from every hole or not. But still, the processing of the tomograms was partially manual, and so we're surprisingly bad in performing automated processing of field series, like we would do data processing for single particle cryo-EM. And several workflows have been developed and published by groups, and we tried some of them, and we were not very successful in that. So roughly 5 years ago, we started developing our own thing. We also looked at the landscape, and then we broke down the subtomogram averaging workflow into blocks, and essentially each block is usually performed by one software that does it really well, and then you can have some scripts to go from one software to another, and then you can do this in certain order, and then you can have certain operations in the order that you can slightly vary. So the manual steps are tilt series alignment, which is surprisingly unperfect, and then particle picking, so identification of your particles of interest in tomograms, is generally not automatic yet, and the reason for that is just the versatility of cryo-electron tomography as a tool, because you can look at a lot of different things, not only of purified protein when you have just one type of protein in your sample, and essentially this is just naturally more difficult to automate. So as a result, there is a lot of manual work to go from data collected, stored in your microscope, to the final structure and classification. So we, Nikita, in my group at the Max Planck in Frankfurt, developed what we now call TomoBEAR. It's the basics of electron tomography and automated reconstruction. So what we're trying to do, it's all open source and it's written in MATLAB, and it calls different commonly used cryo-electron tomography software with a lot of layers of code between it for compatibility and flexibility. And so we managed to do a lot of good default presets. And then we're built it around iMod and Dynamo. So iMod is very good in tomographic reconstruction. Dynamo is very versatile when it gets to subtomogram averaging. It may not give the highest resolution, but it is very good in managing data on the way to high resolution. And then recently, Daniel Castano-Diaz, we didn't do this, it's his work. He released a new tilt series alignment method, which is very robust and has internal controls, which in our hands actually almost worked well. And when the advantage of it is when it fails, it gives us an error message. Right? So we really liked it because this is what you want for automation. If it works, then it gives you a success success mark. If it doesn't, it gives you a failure. Right. So, and then the whole design of this workflow was to not to optimize one step, but to optimize all the steps together, and that worked reasonably well. Several people are working on this now. Our team picked it up in my new lab. It's on GitHub and it's open for alpha testing. We're writing a preprint about this, but we would be happy if somebody would also pick it up and tested it in their own processing environment. Also, once you have a workflow, you can have live data processing functionality. So when you are on the microscope and have a new project, you don't know if your protein is there or not, or if your bacteria are feeling healthy or not. You can actually do a tomographic reconstruction rather quickly and see if you have your object of interest in your tomograms. Right. But we can improve on live functionality later. But first things first, right? So this is what generally a user would do. We would specify a JSON file and it would have blocks. So general block just tells you where files is stored, like what's the tilt angle order, and then what tomograms do you want to have in terms of binning, and then some blocks basically go without parameters because we think that we have coded good presets and it usually works. We also have a wiki and tutorials that give you like essentially typical workflows that you would want. And then there is also a file with the defaults that you can crack and modify it. So in this example, for example, it runs up to a tilt series alignment with Dynamo, and then it converts them to an iMod project. So we have the parallel iMod project because I think that people are very used to iMod, and this includes people in my group. So for example, if fixing alignment parameters is easy to do in iMod. So once that's done, tomograms can be already CTF corrected, reconstructed without human intervention. And also we made a template matching, put it on GPU. It's inspired by Dynamo. So what we do, we allow users to get essentially a EMD deposition, just pull it and then convert it to the proper pixel size and all, run template matching and extract the particles. So basically steps up to here would give you results of the template matching. So you would have your particles, and the particles will have the initial orientations. This is a little illustration. It's again reality interceptor. It's a slice through a tomogram. It's without any pre-processing, so that you can see how noisy it is, but it's actually a nice tomogram. A template, very, very low resolution. So you don't need to have high resolution. You would actually want to be on the conservative side, and you do this all in a very low binning, right? And this is, you basically calculate a cross correlation map. There is nothing new. We didn't just invent this. It was done first time by Hilas Rankakis 20 years ago. Obviously, the peaks that are high, so the user can look at the peaks, and if the peaks are in the position where the particles are expected, then they can be extracted. So we basically look at the histogram of cross-correlation values. And and then once we do that, a lot of garbage is also being extracted together with proper particles. For example, all the gold beads contamination grid edges will also be corresponding to a high value of cross-correlation, and then we would need to do several rounds of cleanup. And then this we can still do automatically, and this is the block that allows a Dynamo alignment project to be created with 3 classes and so-called noise traps. So we have one class that is a real class, 2 classes that are noise particles, and then we will essentially run a couple of rounds of iterations, and then we'll throw away bad classes and continue with the good class. Okay, so essentially up to here, the processing is very streamlined, and the only thing this user needs to do is look at the occasional intermediate results. There are also other ways to pick particles, because template matching typically works for only very large particles. And then if the peaks are not very well defined, there are other options like manual picking. So TomoBEAR interfaces with Dynamo very, very well. And there is a functionality of Dynamo catalog where you can pick particles manually, but also, for example, you can have support geometries where you can draw a vesicle and then parameterize it, so triangulate and extract a particle every, for example, 5 nanometers with the initial orientation perpendicular to the membrane. And then by classification, by rounds of classification, you can classify patches of membrane with your protein or without it, right? And so essentially when you do template matching or geometry speaking, you would also not only have the positions of the particles, so you can extract the particles, but you can also know the initial orientations of the particles, and this saves you a lot of computational time when you do a fine averaging of your particles to an average, right? So with benchmark TomoBEAR, we recorded a data set of purified human apoferritin. So we did commit a cryo-EM crime. We committed a cryo-EM crime. So from 60 tomograms, we could do processing almost manually, almost automatically without manual intervention. And the only intervention was basically importing the results of the pre-processing and template matching into Reliant 4. And then Reliant 4 obviously requires a little bit of manual intervention, but this is all, this is something that the user should actually do. And then we almost went to Nyquist, so this means that we do not damage our data when we do automated processing. It took roughly 1 week, and I was actually rather happy because if you get 3x construction in a week, that's pretty nice. And then we reprocessed our random receptor data set from 2000, from the 2020 paper. It's 63 tomograms, and again, it took a week. The resolution even improved slightly, but I don't think it's very significant. And the only human intervention was class selection and minor refinement of gold beads. So in both cases, most of the time of the processing time and also of the human time was spent on the steps after the particles were already extracted. So roughly, to break down this, both data sets took 2 days to pre-process and 5 days roughly to do subtomogram averaging with Reliant. So Reliant gives beautiful maps, but it is slow, and it's rather resource hungry. So one thing that we realized is also, like, the higher resolution you go, then your pixel becomes smaller and smaller, and then the size of your box becomes smaller and larger and larger. And as a result of that, the processing is just it becomes very, very slow. So Ricardo Sanchez in my group in Frankfurt, my group in Frankfurt finished, and Ricardo went to EMBL in Heidelberg. He has been developing an optimized version of subtomogram averaging. So when we do subtomogram averaging, the first thing we do, we actually record a stack of projections. It's a tilt series, and then we perform 3D reconstruction, and then we end up with a tomogram. So a tomogram is large, 3D, and then in the 3D tomogram, we identify our particles of interest, extract them, called subtomograms, and then we rotate one or more than one reference in order to match them in 3D. So this 3D match is performed by a combination of rotations and Fourier transforms. So what you end up with in the end is a cross-correlation volumes. And when you have, you basically maximize the height of this cross-correlation peak over all the rotations. So once the size of the box becomes very large, it becomes very, very expensive. And then obviously you minimize the peak, so you can do this. It's rather easy. So what Ricardo introduced is to replace the order of operations in calculating 3D cross-correlation. So we know where each particle is in 3D, and this means that we can find every particle in each of the projection image and extract them in the so-called substack. So instead of the 3D input 3D volume that can be rather large, we will have a stack of 2D images that will contain information about the molecule. So, and obviously from the stack, we can create a substack is where one molecule is. So then you can reproject the reference for a given set of angles, also taking into account the step between your projections in the original tilt series. So essentially, you would have the same angles in the set of your reference projections and your substack. And then you can calculate a set of 2D cross-correlations, which is quick. And then from the set of 2D cross-correlation, you can reconstruct a 3D cross-correlation. But the trick here is that you do not need to reconstruct the whole 3D volume of cross-correlation. Typically, it's mostly zeros, and then only in a certain area of this 3D volume, there is some useful information, and you know in advance where this information is, roughly where the center of your particle is. So essentially, you reconstruct a smaller part of it, and the computational complexity is proportional. Instead of as a third power, it's proportional to the second power of the box size, and k is a number of projections in the stack. So k, when k is less than 40, for example, when k is, for example, 40 projections that you have, and your size of the box is more than 40, you get in the computational speed up. And then obviously you can also do the maximization in 3D. You can make this peak sharper, which Ricardo actually does with the whitening filter. And we reported this as a proof of principle on simple data in 2019 on a conference. So when we compare the execution time is just number of rotations. We see that when the box size becomes very, very large, so you'll get a nice fine resolution on a decently sized molecule, your execution time increases very, very much. And obviously you can see how this line will go further. And this is the same graph in logarithmic coordinates. So Susan, basically computational performance is very, very good. I'll go into a little bit details. Why is that also in a second. So Susan is made of several modules, and because Ricardo wanted to build it from scratch, which he has succeeded in. So first thing is, the first model is the estimation of CTF. So obviously we heard about CTF, and then particles in a thick tilted sample are on actually different heights, and as a result you'll get different defocus contribution to it, and then they would have different power spectrum of when you look at them. So the first step that Ricardo does is resampling them to the same height. This is actually, he didn't invent this. It's also done in the other software packages. But so the second step that he does, I think, is rather neat, is that he tries to estimate the defocus by the so-called linearization procedure. So we know that CTF gives us a little oscillatory function where the distance between the zeros at high frequencies is smaller than at low frequencies. So Ricardo looked at the formula, and then he replaced the variable, and in the new around the new variable, it's actually the spacing between CTF is the same as high and low frequencies, right? And then he can basically take the Fourier transform of that and detect the peak. Obviously, he doesn't do this in 1D, but he does it because you can still have a little bit of astigmatism. He does the fitting of an ellipsoid with small and larger axes, but this is very quick. So he does still need to refine this, so he only does this fitting for the initial detection of the defocus and the direction of the astigmatism. But because the precision of this linearization operation and Fourier transform is equals to 1 Fourier pixels, he still needs to refine it a little bit. So this is an example for the untilted image. It doesn't give much of an improvement, but for the tilted image, this actually gives a significant improvement. Yeah, and so you can see that the power spectrum is being, so the ellipsoid of the actual defocus being fit to this power spectrum. And then after which you can go back to the original data. So the difference in defocus estimation between the reference CTF correction tool and what Ricardo gets is sometimes up to 200 nanometers, which is a significant difference when you convert it to differences in your potential resolution. So for 3D reconstruction, Ricardo, when he knows the alignment parameters for every particle and CTF values, and also how much the sample has been damaged by the electron dose, he does Wiener inversion with an ad hoc SSNR that you can input to Susan, and then he performs a denoising operation. So his denoising operation is a combination of blur, which the user can also vary, and often the so-called an equivalent of a solvent flattening. So you can also combine this in a ratio that can be flexible, and then this would be the denoise map that then goes into the next iteration of iterative refinement. So overall, this is the workflow. So he stores 3 databases: particle information, tomogram information, and information about references. And it's performed in C++, and a lot of it runs on CUDA with NPI. The control can be done by MATLAB or Python. So one of the key things about the high performance computational performance of it is that Ricardo does not extract particles to the hard drive. So every time when he performs subtomogram imaging, he loads the entire tilt series into memory, which modern day GPUs allow you to do that. And then on the fly, he extracts the patches, the substacks that correspond to particles, and then he performs the alignment of those. This essentially doesn't, this way you don't need to store anything on a hard drive instead, other than those little updated databases. But you only need to update the particle database after each iteration. So this makes it actually very, very quick. And obviously it scales very well. The more particles you have in a tomogram, then obviously the more the faster it becomes relatively to if you were extracting the particles, right? So yeah, this is Susan. We benchmarked it on several data sets. One is the purified ribosomes on the from the Volta-Face paper. It's one of the typical benchmark data sets that people test the algorithms. So he got to 8 angstrom with the following steps, right? So you basically can see which steps were made and then how it improved the resolution. So I'll go through this a little bit. So the big difference is that the alignment with substacks is first done with the rigid body rotations, right? So you essentially know the relationship between your projections, and then you run the alignment in full 3D. But then you can enable the so-called 2D alignment when each of the projections of your particle can move individually against the structure. So, and you see that this improved the resolution quite a bit, and then in the end, Ricardo's CTF refinement also improved the resolution quite a little bit more. So basically, these steps probably could be performed iteratively, but so we didn't want to spend too much time on purified ribosomes. The important thing here is that we performed this on the same hardware and compared the processing to the reference quick subtomogram average, and it was Dynamo. But yeah, the feature about it is that it was doing the conventional subtomogram averaging with 3D volumes. And then you can see that Susan actually, so gray is the pre-processing with other software packages that you have to do, for example, to series alignment. This was relatively quick compared to the conventional STA processing, but Susan just performed all the iterations much faster. Importantly, once we start recording large data sets, that the disk storage did not improve, right? So the data set had a size of 33 gigabytes, and the processing folder of Susan in the end, after processing was done, is 65 gigabytes, just double the size, and also the number of files some file systems care about it is much slower than a reference subtomogram average and would be having. So the second data set is a little bit more challenging. It's GAG, the structural protein of the HIV virus. I showed you already. This is one of the structures that actually does go to high resolution. People typically benchmark the algorithms using this data set, and it also gave us a good structure. It matches the structure using M, and M clarity is basically 3.0 angstrom, and it's a good structure. But it was processed in a significantly short time, and yeah, so here we see that it almost went over the Nyquist. So the data was in super resolution mode, and we actually managed to run several iterations with the super resolution pixel. And at this point, the box size was very large. I don't recall the number, but this was much larger than any deposition of a subtomogram average in the EMDB at the moment, which I think largest deposition right now is about 400 voxels square. Right. So Susan is also open for testing, and I promise you that we're going to write a preprint very soon. It has tutorials, and you can get it and test it. So we'll be very happy to feedback. All right. So, and this was my last data slide. I hope I could convince you that the workflow approach for cryo-electron tomography and subtomogram averaging saves a lot of effort without the loss of high resolution information. And I could feel it firsthand because I have just moved my labs from Frankfurt to Berlin, and only one person came with me, which was Vasily, the one who did the 4 angstrom structure of ryanodine receptor. And so everyone else started from scratch, and in just very few time, in very short time, we could all produce tomograms and then subtomogram averages, and this was run out somehow with a large level of automation. And it's very satisfactory for a guy to see that people are not spending months to refine the gold beads, but actually do something scientific. So the conventional processing is still very, very expensive, especially for large data sets. And that's why also I think our contribution here is that we developed Susan, which is modular and significantly speeds up the calculations, especially when the volume becomes very, very large. Right now and in the future, we'll be working on incorporating more machine learning tools into TomoBEAR. We think that basically we have TomoBEAR or other workflows can produce a good learning data sets for further training neural networks. And then, for example, you can train your neural network on 10% of data and run it on 90% of data. That would be very, very sweet. And for Susan, Ricardo is working on better denoising for map production, and we work on better accounting for motions, for group motions of particles when they're exposed to electrons. So here I want to thank you, the organizers, for having me, and so the people who did the work. I have several funding sources, and so people who did the work on RAND interceptor was Vasily, and the first work was done by Wenbo Chen. Nikita Balashev developed TomoBEAR, and he built it, so he already finished. And then Artemy Yushkevich, where is Artemy? Yeah, picked it up here, and he develops it as well, and will help out people hopefully to get it going. Ricardo developed Susan, and so in collaboration with Abe, we collected a very, very nice data set of RAND interceptor. Thank you very much for your attention, and yeah, let's have some discussions.